Another really critical part of learning and memory is neurogenesis. It was previously thought that neurons were really only formed in utero and that it didn't really occur across development and into adulthood, but they've since found that neurogenesis does occur in the mammalian brain throughout life, but it just occurs in more specific places. So some of the primary regions of neurogenesis include the olfactory bulb, hippocampal formation, and possibly some regions of the neocortex. The hippocampal formation is particularly important for the formation of new memories. And here we'll watch a brief video on the topic of neurogenesis.
Neurogenesis is the production of new neurons in the brain. For a long time, it was believed that the brain didn't produce new neurons after adulthood. But about 10 years ago, there was some evidence that indeed the adult brain does produce new neurons. There were these new neurons in a part of the brain known as the hippocampus. And the hippocampus is a part of the brain that's involved in learning and memory. We know it's necessary, actually, for some aspects of learning and memory. After much discussion about these new neurons and the fact that they happen to be in the hippocampus, we decided to investigate whether or not they might potentially be involved in learning and memory. So over the past 10 years, we've accumulated considerable evidence that they are, that they're certainly affected by learning, there's no doubt about that, and they might even be necessary for some aspects of learning. So the hippocampus has these cells that are somewhat like stem cells, and so they just continuously produce these new cells. So these cells are very, very sensitive to their environment, the external environment as well as kind of the internal environment of the animal, hormones and things like that. However, once these cells are produced, oddly enough, most of them don't survive. What we wanted to know is whether or not if we could keep these cells alive if animals were to learn something new. So what we did was we train animals on these different learning tasks that require the hippocampus, that depend on the hippocampus to do it, right when the cells are about to die. So the cells are generated, and so we mark them with a neurochemical marker that can detect these new cells. And then we can monitor them across this period of time. And then we train animals on these various learning tasks. And we find that the animals that learn this task keep these cells. But if you don't learn the task, the cells die. We use the rat because we know a lot about the hippocampus of the rat, and so we can manipulate these various learning tasks and determine, what is it specifically about learning in that model that rescues these cells. The tasks that we focus on are called associative learning tasks, where an animal learns to associate one stimulus with another stimulus later in time. The task that's really effective is a task known as trace conditioning. And in trace conditioning, the animal hears a tone, and then there's a period of time where there's nothing. And then half a second later or so, he receives the stimulation of his eyelid, and that causes him to blink. It's called eyelid conditioning. And the animal learns that the tone predicts the stimulation and blinks eventually in anticipation of the eyelid stimulation. So it's not the only type of learning that rescues these cells, but that's just the one we've mostly focused on. One other focus of my laboratory has been sex differences, looking at sex differences in learning and how those sex differences affect the brain. When we were trying to determine whether or not it was really learning, that rescues these cells from death and not just something about the training experience or the stimulation or even the stress of it, we thought, well, maybe it would be interesting to look at sex differences in learning, because we know that under certain conditions, females learn better than males do. And so we thought, well, it would be interesting to see if then, by virtue of the fact that they learn this task so much better, would they rescue more cells than males do? And indeed, that's what we found. I think, that these data that we have in animals and rodent species are maybe suggestive and maybe support this idea that if you continue to learn throughout your life, you are continually changing your brain and potentially keeping neurons there that wouldn't otherwise survive.
So I thought that was a great video to talk about the role of neurogenesis in the context of learning and memory. And so now I have a brief question that is going to be posted as a participation question for the class. Just one of the participation questions for this week. And this will be a brief blog question. What are some of the implications of neurogenesis as a function of associative learning? So at the time that I post this lecture, I will also post this participation question. So please, if you have not seen it, then look for it. And after you are done, you should be able to see some of the responses of your classmates, and then you can just look over what they said versus you said, and hopefully there will be some interesting associations or implications that people discuss. All right, so another important aspect of learning and memory is the role of the environment in brain plasticity. So we talked about how critical brain plasticity is, this idea of that your brain is changing. And this changing brain, the way that it changes and whether it changes in adaptive versus less adaptive ways, is going to be highly influenced by the environment. An enriching environment is very, very important for brain health and growth. Raising rats, for example, in enriched enclosures is associated with increased brain weight, more astrocytes, more synapses per neuron, more dendrites, more blood capillaries, basically an overall better functioning cognitive neural system in the brain. And here we have another class question, and we have more class questions at the end of the semester just because you guys have now garnered a fair amount of material and can actually think about some of these more complex questions. So, if there is a really strong link between enrichment and brain health, then what would the link be between enrichment and SES, which is socioeconomic status? What are some of the downstream implications of possibly having a lower enriching environment from lower SES? So I want you to think about this, and again, when you have some time, fill out the participation question that will be posted as part of this week's lecture series. I also recommend, before completing the participation question, to listen to the rest of this lecture. To give you more of an idea of the role between enrichment and neural function, we can just speak more generally about the role between experience and neural complexity. So here's an example of a neuron in our finger area, and here is an example of a neuron in our trunk area, and here's an example of a neuron in our language area. So the complexity of these, the area, our trunk area and our finger area, they largely have a similar structure, but there's a bit more branching here, so a bit more opportunity for this neuron to communicate. And this really is in line with how this neuron is going to be active more, it's going to be used more, and so when you experience things, you're going to be using this finger area neuron a little bit more than this trunk area neuron. So the development and shape of this neuron is going to reflect that increased complexity. Down here, you see a neuron that's in the language area, and this is even more complex. A language, of course, is highly complex, highly associative. So when you engage in any sort of language processing, then you're engaging in multimodal processing of information. There's going to be verbal information, there's going to be visual information that corresponds with that verbal information. You're going to be interacting with another person. So the complexity of this neuron is going to reflect the experience that the body would go through when it engages in language processing. There are also a number of other factors that affect plasticity. Hormones, trophic factors, and drugs can all affect neuroplasticity. So high levels of estrogen, for example, is associated with more dendritic spines in the hippocampus. And if you listen to that brief research spotlight that I had put that was presented as part of this lecture, talks about how females learn some content better than males. And this might actually be reflective of that females tend to have higher levels of estrogen, and that this higher levels of estrogen is associated with more dendritic spines in the hippocampus. In contrast, lower levels of estrogen is associated with more dendritic spines in the neocortex, but fewer dendritic spines in the hippocampus. This could be related to memory age decline in middle-aged females. So there's a number of possible reasons for this. But on the whole, this relationship is meant to demonstrate the fact that hormones do actually affect plasticity in the brain and the way neurons interact with each other and how complex they are in their connections. We see a similar effect for testosterone. The low levels of testosterone is associated with more dendritic spines in the neocortex. So here's an example of how these dendritic spines can look different. So here we have high estrogen levels on the left, and you can just see that there's more dendritic spines, and low estrogen levels on the right, and you have fewer of these dendritic spines. More dendritic spines allow this neuron to communicate more with other neurons, and fewer dendritic spines means then, again, that a neuron has lower connections with other neurons. Another hormone that affects plasticity is glucocorticoids. Glucocorticoid is released from the adrenal cortex in times of stress, so it's highly important in the stress response of the body. Glucocorticoids also assist in the metabolism of proteins and carbohydrates and the control of sugar levels in the blood and cells. So glucocorticoids overall ready us for times of stress. They make sure that our body is prepared to deal with potentially threatening and stressful things. And steady levels of glucocorticoids are seen with prolonged stress, and these actually may be neurotoxic. While glucocorticoids are important for stress, if you were to have steady high levels of glucocorticoids, that is actually toxic for neurons. So the type of steady increase that you would see in glucocorticoid response would occur in the instance of prolonged stress. And what they found is that this increased stress response can result in an increase in glucocorticoids that, of course, while beneficial in the short term, over the long term is not just toxic to hippocampal cells, but it's also toxic or can result in other physiological problems that can increase weight gain and make stress harder to manage. And what we find is in really intense, stressful situations that hippocampal cells die. This can impair memory. And so in some respects, you could see that this would be beneficial for in the short term, that if you have a highly traumatic, stressful event, then you may not actually want to remember it as much. The problem is that when glucocorticoid levels actually stay high over a longer period of time, our bodies are not designed for this steady, long increase in glucocorticoids. And so over time, something that is beneficial in the short term can actually become toxic. So it's not just that stress and sort of like, oh, you've had some sort of stressful circumstance, that it really is long stress over long periods of time that can greatly impair your body's response and can also result in the death of hippocampal cells that can make it actually harder to engage in the formation of newer memories and mood repair. Neurotrophic factors are also highly important in plasticity. The nerve growth factor is a neurotrophic factor that stimulates neurons to grow dendrites and synapses, and in some cases it promotes the survival of neurons. The brain-derived neurotrophic factor, known as BDNF, may also actually enhance plastic changes, such as the growth of dendrites and synapses, and this is increased when animals learn to solve problems. So this suggests that some of these neurotrophic factors, nerve growth factor, or brain-derived neurotrophic factor, may actually facilitate change in neurons, specifically dendrites and synapses. And so this change can promote survival, but it can also promote growth so that we can learn new things. And this also relates back to the video that we presented, in which actively engaging in learning and experiencing your environment and forming new associations is going to be healthier for maintaining your brain's function. There's also a range of effects that drugs, particularly psychoactive drugs, can have on plasticity. So you have behavioral habituation, and then you also have behavioral sensitization. Behavioral habituation is a decrease in sensitivity to the repeated administration of a drug such as alcohol. Habituation is associated with a decreased number of receptors and reduction in synapse activity. So behavioral habituation, if you were looking at this perspective of how change in learning increases growth, habituation is kind of the opposite of that. So not all habituation is bad, but some behavioral habituation, it basically can result in stagnancy, sort of neural stagnancy. It's a lack of change. And if you are not learning new things, you are not forming new associations, then you don't have that stimulation that would promote the formation of newer dendrites and would promote synaptic activity and promote or lengthen cell life, particularly in the hippocampus. The type of habituation that is associated with addiction can be particularly problematic because your body is constantly trying to maintain a status quo. So actually, if you have too much alcohol, then of course alcohol affects your body through neurotransmitters. So if you have taken alcohol and you have an increase in a certain amount of neurotransmitter, then your body is going to again try to actually return to baseline and reduce aspects of that neurotransmitter. And it's going to reduce it by reducing the number of receptors, or reduction in synaptic activity, or reduction in dendrites. And then in that reduction of receptors, dendrites, or synaptic activity, then you're going to need to drink more alcohol to achieve some of the behavioral effects that you would want to. And again, your body is going to try to maintain the status quo, and it's going to further reduce the receptors' synaptic activity. So overall, over time, the subtle changes that are part of habituation will also set up a circumstance that increase the addictive potential of certain drugs. We also have behavioral sensitization. Behavioral sensitization is escalating behavioral response to the repeated administration of a psychomotor stimulant, such as amphetamine, cocaine, or nicotine. It's also called drug-induced behavioral sensitization. Sensitization is also associated with an increased number of receptors, synapses, and dendrites. These changes that occur are localized to regions, for example, the prefrontal cortex or nucleus accumbens, that receive a large dopamine projection. So basically, after you have psychoactive drugs, you can habituate to drugs, of course, but then you're also going to have a sensitization response after you've taken some drugs. So this can be how you would find yourself in a drug-induced state, being highly, highly receptive or stimulated by certain stimuli. And this is something that can be positive, that people would seek in a drug-induced state, that they would take more drugs to achieve. This is, again, a psychomotor stimulant. There are certain drugs, amphetamine, nicotine, that happen all the time, and they try to, again, they sort of increase our arousal. So in increasing our arousal, then we have an heightened response. We are more sensitive to certain things that we encounter in the environment. So this can result in an increase in receptors, synapses, and dendrites. So this can result in making us more prone to change, but these changes can be localized to regions primarily associated with dopamine, and they can still be problematic because you can have this increase in sensitization that you seek, so you want to be stimulated after you have coffee or smoking to a certain extent, but then after a while that sensitization goes down, and so you have to again take another cigarette or have another hit of something. In this way, both behavioral habituation and behavioral sensitization can affect drug addiction. So we've talked a fair amount about brain plasticity in this chapter. Overall, there are some guiding principles of brain plasticity that's important for you to know and understand. One, behavioral change reflects change in the brain. So we've talked about this a bit, that when you engage in any sort of behavioral change, then that is reflected in physical changes in the neural structure of your brain. All nervous systems are plastic in the same general way, meaning that anything can be changed. Our brain is not set in a permanent fashion. We are constantly changing and adapting. There are also plastic changes that are age specific. So there are certain changes in plasticity that only occur at developmental stages. Prenatal events can also influence brain plasticity throughout life. So what happens in the utero can influence how your brain processes information for the rest of your life. There are certain hormones that influence organizing principles while in utero, and prenatal events can influence the release of those hormones. And so in this way, you can have an event that happens while in utero that has organizing and deterministic effects on who you are for the rest of your life. Plastic changes are also brain region dependent, so the brain does not change universally across all the different types of brain regions. Some brain regions change more and some brain regions change less. The hippocampus, for example, has continual neurogenesis, while other regions of the brain have much less neurogenesis, and other regions of the brain might engage in more synaptic and dendritic changes to reflect learning. Experience-dependent changes also interact, so this is known as metaplasticity. So you have changes that occur as a function of development, you have changes that occur as a function of experience, and you have other types of changes that also can occur as a function of experience. So the different types of changes that occur as a function of experience, these changes can interact to result in this idea of metaplasticity, that experience can affect you in multiple ways, and this experiences can be additive, where the changes are building upon each other, or they can be quite distinct. And finally, plasticity has pros and cons. Plasticity can be very good in certain contexts in terms of maintaining cognitive activity, maintaining neuron health, but then plasticity elements can also be problematic in certain contexts, such as those associated with drug addiction.